Okay, thanks very much. The work that I'm going to be presenting today looks at how decision-making computations might differ across different subregions of macaque prefrontal cortex. It's joint work done together with a PhD student, Nishmalala Sekera, in Steve Kennelly's lab at UCL before I moved to Oxford a few months ago. Now, the way that we often study value-based decision-making in the lab is quite straightforward. A subject might be asked to lie inside a scanner, a fixation point would appear on the screen, and then two food items would flash up on the screen, and the subject would choose one of these two food items, the one that they prefer, with a button press. And this is kind of decisions, of course, what we think of as two alternative forced choice. And what's been found is that by regressing the values of various items against neural activity, it's been possible to find various regions of the brain that correlate with these values when making such a decision, most notably in frontal cortex. But a challenge for some of these paradigms comes when we start to think about how these decisions might play out in the wild, decisions a little bit like this one. In the supermarket. And although they might superficially seem quite similar to the one that we just saw in the lab, there are also some things that the lab experiment clearly hadn't captured. So the person seems to sequentially deploy his attention backwards and forwards as he's deliberating, weighing up the advantages and disadvantages of each of the different options. There might also be examples just like this one, where he might evaluate just one of the items on offer and decided that he liked it so much it wasn't even worth deploying attention to any of the other choice alternatives. And so this might give us pause for thought as to whether those paradigms and models which have been used in those paradigms really capture all of these different processes, particularly the interaction between attention and decision-making. How does this person decide where he's going to look next? How did his shifts in gaze affect the eventual decision that he might make? How does he decide when to stop sampling information? And most importantly, for the point of this talk, what are the representations within frontal cortex that support these various processes? Okay, so to address some of these questions, we trained two macaque monkeys to perform the following decision task. So subjects were trained to choose between two options, positioned on the left and right of the screen, each of which consists of two pictures. And the pictures provide the subject with information. One picture is going to tell the subject about the amount of juice that will be available if they were to choose that option, and the other picture tells the subject about the probability with which they'll receive that juice. And the pictures are going to be randomly sampled from this set shown over here on the left, and the subject has already learnt the meaning of these pictures very well before the experimental session has even started. So in this example, he's choosing between a 70% chance of getting one drop of juice on the left-hand side versus a 10% chance of getting 5 drops of juice on the right-hand side. But the trick that we play is that the subject doesn't receive all of this information at once. Instead, he has to sample it sequentially. So the first thing that appears is this fixation point. And once he's acquired this, the 4 picture locations are going to appear, and 3 of them are going to be covered up with a gray square, and one randomly selected location is going to be highlighted with a blue square. So that instructs the subject to saccade to that picture location, which reveals the picture that's hidden beneath. And he has to fixate that picture for 300 milliseconds before the trial can continue. And in fact, for the purposes of today's talk, most of the neural data that I'm going to be showing you actually just comes from this very first fixation, the very first time that he deploys attention to one of the two options. But I'll just walk you through the rest of the trials to be complete. So after this first fixation, one of 2 possible things can happen. In half of the trials, another blue square is going to appear on the same option as the first cue. The subject saccades to the square, revealing the picture that's hidden beneath, at which point he knows all of the information, the reward probability and magnitude about that particular option. On the other half of trials, the blue square is going to appear over on the other side of the screen, And if this happens, he's now learned all of the information about one of the two attributes, in this case reward magnitude, but he doesn't yet know anything about the other attribute. And then once those first two pictures have been viewed by the subject, the two remaining locations become highlighted, and from here on in, the subject essentially becomes free to do as he chooses. He can either go ahead and gather the two remaining pieces of information by deploying attention, saccading towards them, or at any point in time, he can terminate the trial by moving the joystick that he's got in his hand to choose the appropriate option based upon the information that he sampled thus far. Once he's made his choice, then all of the four pictures are going to get revealed, and then after a short delay, he's going to get that juice reward probabilistically delivered with the reward magnitude of the chosen option. Okay, so we recorded from two macaque monkeys as they were performing this task, and we positioned our recording chambers so that we were able to access all three of these different regions shown on this slide in frontal cortex. The dorsal bank of the anterior cingulate cortex in red, area 13 of orbitofrontal cortex in blue, and the principal sulcus in dorsolateral prefrontal cortex in yellow. And by lowering multiple electrodes into these regions, we could typically sample somewhere between 10 to 40 neurons a day from these regions, so we can get a reasonably large population of neural responses from each structure. And the analysis that I'm going to focus on today are some ways that we've recently been trying to use to understand the structure of these neural population responses at the time when the first saccade is made, when the first piece of information is being attended. And these capitalize upon a key feature of prefrontal neuronal responses, which is that neurons in prefrontal cortex tend to show a lot of heterogeneity within each brain region in terms of what they encode. And so this allows us to perform a representational similarity analysis to assess how information is being encoded at this first saccade without strong prior assumptions. So RSA will be familiar to most of you, as it's been mentioned in previous talks, but I'll just quickly walk you through it. So the way that it proceeds is like this. We can look at all of the trials where, say, the 0.7 probability Q has appeared on the left-hand side of the screen. And we can do this for all of the neurons in a given brain region, let's say orbitofrontal cortex. And then we can just plot the z-scored firing rate for each of the different neurons as they respond to this cue along the x-axis. So each dot here is one neuron. We can then move to a different set of trials, let's say the ones where the 0.7 probability Q is now over on the right-hand side of the screen rather than the left. And that's what's being plotted on the y-axis here. And now when we correlate those two vectors together, what we find is that there's a positive relationship between those two population codes. In other words, the OFC population seems to be responding very similarly to this stimulus, irrespective of whether it's on the left or the right-hand side of the screen. We can then shift our attention to, say, a different set of trials where a different stimulus is being presented on the left-hand side of the screen, let's say the cue that represents one drop of juice. And plotting this on the y-axis now reveals a slightly negative relationship with the population response for the 0.7 probability Q. Okay, so that's the basic idea. We can then go ahead and repeat this procedure for all of the different possible cues and all of their different combinations, and we can use this to build up a matrix of correlation coefficients that shows how similar the population response is to different cues in different locations. And when we do this in orbitofrontal cortex, we can see this really quite striking structure that emerges from the population responses. So just to orient you to how this matrix is being sorted, each element shows a representational similarity between two different conditions. We've got the 5 probability and magnitude cues being sorted from the lowest value up to the highest value. And we've got them being presented on the left-hand side of the screen here and on the right-hand side of the screen here. So hopefully you can see a couple of things that kind of immediately pop out of this matrix. So first of all, there are these off-diagonal elements here. And these indicate that there's representational similarity in the OFC population for the same stimulus identity, irrespective of whether it's being presented on the left-hand side or the right-hand side of a screen. We can also see this gradient of responses as squares that's repeating itself across the entire matrix. And so what these represent are a value response for the currently attended queue, irrespective of its spatial location. In other words, low-value pictures tend to be positively correlated with other low-value pictures, and high-value pictures will tend to be positively correlated with other high-value pictures, but low and high-value pictures will tend to be anti-correlated with one another. Okay, so that's what it looks like for orbitofrontal cortex, but what about the other regions that we recorded from in this task? Well, if we repeat the same analysis for both the anterior cingulate cortex and the lateral prefrontal cortex, we see a very different pattern of responses emerging. First of all, neither of those two regions seems to provide that strong stimulus identity code that we were seeing in OFC. And in ACC, we do seem to have some evidence of something a little bit like the value code, but interestingly, it seems to take on more of a categorical, block-like structure, as if it's categorizing the queue into, is this an item that I like, one that I might ultimately accept at the end of a trial, or is it one that I don't like, one that I might end up rejecting? And then lastly, we can also see that dorsolateral prefrontal cortex seems to be mostly caring about the currently attended stimulus location, so whereabouts the subject is currently looking. We can also repeat this process across time as the subject is moving his eye to to attend to that cue, to see how these different representations emerge relative to one another. And to try to get a little bit more formal about these different representations, what we've also done is built a number of different templates that we capture each of those different features that I mentioned on the previous slide, and we can regress these back onto the RSA matrices at each point in time. So what you see is that as the subject saccades towards the cue, the template that captures the deployment of spatial attention emerges really quite early in lateral prefrontal cortex, so it suggests that it's linked to the time when the eye moves as opposed to anything to do with stimulus processing. But on the other hand, those other representations that we saw of stimulus identity, value, and the accept-reject code that we saw in ACC all seem to emerge more or less in parallel, peaking around 300 milliseconds after the stimulus is being presented. We can also in this task go on to examine what happens as further cues are being examined by the subject, up to the point when the subject makes a final decision. Unfortunately, I don't have time to describe these in any great detail, but I just want to mention a couple of headline stories. So first of all, in orbitofrontal cortex, but not in other regions, what we find is a signature of attention-guided value comparison as the subject shifts their gaze from one location to the next. In other words, neurons that positively encode the stimulus value that you're currently attending to now negatively encode the value of stimuli that have just previously been attended and are currently being stored in working memory. And then secondly, in anterior cingulate cortex, but not in other regions, what we find is multiple regions that ramp prior to the commitment to making a final decision, prior to moving the joystick. One of these corresponds to a belief confirmation signal when the currently attended cue seems to confirm your current expectation about which option to choose. And the second of these seems to correspond to an action selection signal, representing simply whether the subject is going to choose left or right on the current option, a little bit like some of the neurons that Mike Shadlin was showing in his talk yesterday. So if you want to find out more about these results, you can take a look at our preprint, which was released on bioRxiv last month. And I also want to mention that the raw data and analysis scripts for this is going to become available for download on CRCNS once the paper's accepted for publication. So with that, I'd like to finish by thanking some of the key people who were involved in this work. Nishmalala Sekera, who is a PhD student who is the kind of driving force behind the data collection on this project in Steve Kenneally's lab. Archie de Burker, who's in the audience, who's now at Element AI in Montreal, and first had the idea to try out RSA on this data set, and also Tim Behrens, Simon Farmer, and Bruno Miranda, and the Wellcome Trust for funding. Thanks so much.
We have time for one question.
That's a great question. The question was the difference between VLPFC and OFC representations. I can only speculate. I guess one well, I won't speculate because I don't really have a good answer. One thing that maybe I will mention is that we have been trying to also compare OFC and VMPFC representations in this task, partly driven by the fact that VMPFC seems to so often code value representations in human fMRI data, and VMPFC generally seems to just look like a kind of weaker version of OFC so far. So we haven't been able, and that seems like something that's quite often seen in neurophysiological studies. It's surprising how hard it is to see representations in VMPFC. So sorry that wasn't a question you asked, but I don't have a good answer.
All right, let's thank the speaker again.